So you can see it now?
Yes.
There you can see the talks are given here, so.
Yeah, good. So I think the first one would be by Elena.
Yeah.
Elena, would you like to give like 1 minute general explanation of your presentation, and then people ask questions?
So, okay. Elena presentation was about the changes of the Crimean saline lakes. There are a lot of the saline lakes in the Crimea, and long-term changes occur during 2 centuries due climate variability and anthropogenic interventions. And if some lakes reflect climatic variability fluctuations, another is more impacted by anthropogenic press. And it's difficult sometimes separate each cause main, but we can conclude that, as usually, climatic variability is semi
Okay, yeah, thanks, Nickolai. I think there is also a little bit of audio problem. So is there any questions to Elena?
Yeah, I have not good contact with Elena.
Elena,
Yeah,
are you here? Are you here, Elena?
No, some problem with connection. He called me and said,
okay, so
I
maybe
don't know what.
Yeah, so maybe we can go to the next one, which is
There's
Vanya.
we could take a discussion
Sorry.
about it, because we could combine it also with Nickolai's own presentation. Because Nickolai, you talk about alternative stable states in your talk,
Yeah.
and using these Crimean lakes as an example of this.
Yeah, but
But I understand. I have several questions now. But the question is that you have in your Crimean systems that Elena presented was that you have this channel. So you change salinity from high to low, and later it goes from low to high again. Did you see?
Yeah.
Did you see alternative states there, or was it a gradual shift in the community? Was it a...
I see. As usually, we have not monotonic changes. It's also it's abrupt changes. But this tipping point is different than growth salinity or it's decreasing. But we have changes without abrupt only in small salinity intervals, because in our lakes the salinity changes very high range. As example, in some lakes from 10, 20 gram per liter to 300 gram per liter. And in this case, we all time observe abrupt changes. It's okay? Or you need to ask more?
My point was more this, that with alternative stable state, you can have a shift, you can have abrupt shift completely without alternative stable state. It can just be that you reach a threshold in salinity, and then, for instance, fish die and it changed completely. Or you get from phytoplankton to benthic community because of salinity change. So the best you could say, test of your hypothesis of alternative stable state is to see that when you increase salinity, as you had in the case, in some of your case studies Elena told us about this, an increase in salinity and you have a decrease in salinity. Do you then confirm your alternative stable state idea, or is it just a shift occurring when you reach the threshold in salinity?
Yeah. If we look on salinity changes and the proportion between zooplankton and zoobenthos, we can see that after some salinity, all benthic animals shift to planktonic lifestyle. And it's mean that full changes of trophic chains in lakes.
But
It's one of the examples. In some salinity, we have, as example, phytoplankton, and then salinity grow, and we have green filamentous algae mats mostly, and after continuing growth, some salinity we can see again phytoplankton, but only as usually with Dunaliella.
Yeah. Well, but I should not continue. But that's the question is, do you see resilience? This means that if you are in a plankton system, when you change salinity, does it stay in the plankton system longer than you will expect? Because that's the alternative stable state. Else it's not alternative stable state. Else it's just salinity shift and threshold shift when you pass the salinity. So, but you have good data for that.
No one alternative state. It's some salinity can be different states, and it's related to some other factors also. As example, wind and the input of organic matter and biogas, etc. It's not a strong relation salinity and point of abrupt shift. There is some range there we can see abrupt changes, shift.
Yeah, but regime shift is not the same as alternative. I start with, I think I think we can discuss that another time. I should not take the time. So other question from people.
Yeah, okay.
Okay, just a second. Thank you very much, Eric and everybody. I just would like to say something. So there are 7 more participants, right? And we should calculate about 9 minutes or now 8 minutes for each. So Nickolai, can you control more or less the time so everybody has time to speak? And maybe it's a good idea also to follow an order. So perhaps Vanya can talk now and then Juan and so on, with about 8 minutes or so for each. Otherwise, we run out of time in the end and some people may not speak. Thanks.
I think it's okay. 7 minutes per presentation. And
okay.
Who is next?
So according to this is Vanya.
Okay.
Vanya Milankovic, is she here?
Milovanovic, it's okay.
Oh, sorry,
okay, no, it's okay. Okay, well, hello everyone. The name of my presentation is Abandoned Artificial Saline Pond, a safe place for rare or endangered species. So our results are just preliminary results obtained after only one sampling occasion. So we investigated the biodiversity of macrophytes, phytoplankton, and phytobenthic communities in one banded artificial saline pond, and we concluded that it represents a safe place for rare or endangered species. Why? Because here we found a stable parthenogenetic female population of Hara canescens, which is important because this species its populations are isolated, and it is worldwide considered to be threatened, endangered, and rare, while in Serbia it has been declared to be probably extinct in the wild. And this is second currently known locality of these species in our country. When we talk about phytoplankton community, we detected 27 algal taxa belonging to 5 divisions, and we detected low diversity, which is not surprising considering the fact that the species diversity is generally inversely related to the lake salinity. And in phytobentic community, we detected 15 diatom taxa, and the most dominant genus was Nichia with 4 recorded species. And as a conclusion, we can say that generally low diversity, typical for saline brackish habitats, was detected in relation to all communities. However, some typically brackish, rare, or endangered species were recorded. And so we propose conducting a detailed study of this and other ponds of similar origin. So that is all.
Yeah, so thanks, Vanya. Are there any questions for Vanya? You can also write down at the chat.
I can ask a
Do you
question.
have some questions? Right, please.
May I ask a question? Can you hear me, Vanya?
Yes, I can.
Thanks. It's a nice work that you are presenting. I just wanted to know what is the salinity tolerance of this species, this rare species that you found. Is it
Well, it is typically brackish water species, but I do not know exactly the answer to your question. I'm so sorry, this is my first conference and I am very confused.
No, don't worry, don't worry. Thanks a lot, it's fine.
Thank you.
I have a question also. How many ponds lakes are you going to study? Pond and lakes are you going to study? You say you have taken this one, but there are many others. What is the plan for the future?
We studied also another one, Plava Banja Pond, and Hara canescens was also found here. But there are several more, but we have to find funding for this to study all of these ponds. Thank you.
Next presentation, it's Dr. Sarma. Oh, no.
No, yes.
Oh, excuse me. Jean Soria, yeah. Welcome.
Okay, I am here. Please, Eric, can you stop the share of your screen and I can put a small presentation?
I think it's Eric who is
Okay,
sharing.
okay.
Good.
Okay, I'm Juan Soria. I'm from the University of Valencia. our study is about the monitoring of submerged aquatic vegetation using remote sensing imagery in Gallocanta Lake. Gallocanta Lake is a saline endorheic lake in Spain, in Aragon, and has a wetland around the lake. The lake is in the center of the wetland, and the wetland is surrounded by many fields and mountains. We have studied before the hydrologic regime of the lake, and now we study the vegetation in the wetland and in the water of the lake. We try it with sentinel imagery, and first one found some images without clouds, mark the area of the wetland of the lake, and apply one index to study the vegetation. We try some indices, the NDVI, the NDV45, and also the best for us was the NDI45 that shows better the vegetation in white color in this picture and no vegetation in black, in gray and black. The other indices are not also as clear as this one. The result is that we can monitoring the vegetation in this scale. We found in blue colors no vegetation, in yellow small vegetation, and when the green is dark are more vegetation, not only in the water, also in the wetland. We have masked the out of the wetland, and we see only this the RGB image, and over that the covered area of the vegetation. We see that in this one we have only vegetation in the wetland and no in the lake, and in this one we have in the lake and in the wetland. Around the year 2019, we can see how the vegetation is more dense in the center of the lake, and the case in autumn, and in winter there is no vegetation in the wetland and a small quantity in the water of the lake. And for 2020 we have the same as vegetation covered all the area and the lake, the whole lake, and in autumn disappears from the wetland, only rest in the lake, and when the snow falls and frozen, all the field, all disappear of the lake. Is that the remote sensing is a good tool to see if we have aquatic vegetation in all the area, or not only the wetland, also inside the water of the lake, because the water has clarity, and we can see the plants in the bottom of the water and emerging in the surface. If you have some question about, I can answer, or want to place in the chat. Any question?
Oh, I want to ask, do you have floating mats, algae mats in your lake?
Sorry, I can't hear clearly. What do you ask?
Do you have filamentous green algae floating mats in your lake?
Yes. Yes, we have in some periods, we have filamentous algae in the water also. More, not only hydrophytic vegetation, also small filamentous greens. I think, I don't know what is the species, but not sure if it's green algae, green blue, or only green algae.
And
This is
is it
a
possible?
very saline lake. We think that when the salinity is more than 50, vegetation decreases.
In such salinity, we found rupia and tlasopra only. Are there more questions?
I wanted to ask a question, Juan.
Yes.
I just want to know
Hello,
whether that.
Dandiri.
Hello, how are you?
Thank you. Fine.
Yeah, I just wanted to ask you if the differences are because were seasonal, or in the different years also you found some differences. And so did you find any specific difference in 2020? Is this lake a very touristic area?
No, this is no touristic area. Do you ask?
Yes, this is not
No,
touristic.
this is not. Only rain fields. No people. No.
Okay.
I think that there are only in the two villages around the lake, only 100 people living here. This is the
Yeah.
the empty Spain. Yes.
No, and also if the differences are seasonal or are they between different years? Interannual differences.
Interannuality. Yeah. We found that the vegetation disappears when the lake is drought. The lake every 5 or 7 or 3 years in summer dries all the lake, and all vegetation disappears. But it will appears now when it's flooded with fresh water. The fresh water redissolves the salt and turns to a saline lake, and when evaporating now turns to very high salinity, and we think that then makes the disappearance of the some vegetation.
Okay, I think Nickolai has asked us to stop.
Yes, I have a question in the chat that if we calibrate the vegetation with field data. Yes, we have some field data, not more, only 3 or 4 times, but we have measures in situ of the coverage of the vegetation. It's easy to see because the water is transparent. We can see the bottom of the lake. And when it's vegetation, we see clearly where is the plants in the water. It has 1 or 2 meters of water only. The deep point is only 3 meters, and only in one small corner. In the whole lake has low, shallow lake.
Do you have more questions?
Yeah, I think there is one in
about
the chat.
the modeling. Yes, we are now trying to form a correlation between species, the private species in the water, and the image of the satellite. Yes, this is a second work that we can make in future studies to see if we can identify the species from the satellite also.
And can we use separate wetlands vegetation and vegetation in lake.
No, yes, we know where is the wetland. Previously to the work, we know where is the wetland and where is the water of the lake. And we make a mask to identify where is always wetland, only vegetation, and never has water, and where is the area that can be covered by the water of the lake when it's full of water. Yes, we have a mask. The wetland is not covered by water, it's only wetland.
Yeah, I see.
Okay, so
Other
good.
questions?
So,
Yeah. If no, thank you, Dr. Soria.
Thank you very much for the attention. Hello, Nandini. See you.
Who is next?
it's Nandini. I think it's you, right?
Yeah, thank you, Andros. Can I share the screen for just a moment?
Yes, sure.
Okay. I'd just like to explain very briefly what we did in this study. Can you hear me?
Yes, I can hear you.
Okay. So we looked at the demography of this rotifer, Hexarthra jenkini, which is commonly found in saline waters, from two different habitats around Lake Texcoco, an ephemeral and a permanent habitat. Lake Texcoco was once part of a very big water body in central Mexico, which had freshwater and saline systems. It is one of the few soda lakes that we still find over here. The Aztecs even harvested spirulina and included it in their diets. So this is Lake Texcoco. We just have remnants of this lake. And then we have Lake Zumpango in the north and Lake Xochimilco in the south, Lake Chalco also. But much of this has disappeared, as you can imagine, for a city of 30 million people. And this area was being considered to construct a new airport. Fortunately, they have given up on this idea and the habitat remains. So Lake Texcoco has permanent basins, which is the Nabor Carrillo area, and it has very shallow turbid pools that dry completely in the summer and refill during the rainy season. There are several zooplankton taxa that you find in this lake, including different species of rotifers and cladocerans and copepods. So considering the very temporary nature of these pools, we thought that the species that, if it is the same species, Hexarthra jenkini, from the permanent and the shallow water body, the temporary water body, the species which are there in the temporary water body should have higher growth rates. And that is what we tested in this study, and shorter lifespans. So we conducted the study using nanochloropsis, as you can see in the video. And what we found was that the strain from the lake had lower growth rates than the strain from the pool. The pool strain had very high growth rates, and which allowed them to recolonize and utilize the resources in these habitats rapidly. They are also more tolerant to salinity, as you can see in the egg ratios. And similar studies have reported results, as we have found, of Hexarthra jenkini. However, there are differences in the strategies of Hexarthra jenkini from different water bodies. And for instance, we conducted a similar study from another saline lake in Mexico, Lake Alchichica, and Hexarthra jenkini reached much higher densities there in laboratory experiments. So we need to look at the possibility that we are not working with cryptic species in this case. Thank you very much, and any questions, I'll be happy to take them.
Other questions? Please ask.
Hi, Professor Nandini.
Hello.
Hi. I want to know if you have detected the presence of resting eggs or something in the experiment.
Yes, we found resting eggs, and if you take the sediment from this area and hatch it in the lab, several species hatch. But resting eggs in Hexatra were fewer than resting eggs in another species that we have tested from the same water body, that is Brachyonus dimidiatus. Brachyonus dimidiatus produces many more resting eggs than does Hexatra jenkini. But resting eggs are produced, and that's how the egg bank helps to re-establish the system whenever the conditions are favorable.
And do you know if there's a difference in the rates of resting eggs production between saline and freshwater systems?
No, we didn't test freshwater. Hexatra jenkini is normally found in saline systems, but we have not tested whether the rate of production of the resting eggs are different and the rate of hatching of the resting eggs are different between the species which is found in the ephemeral water body, which is the temporary water body, rather than the species which is found in the permanent water body. We haven't tested that. And neither have we checked at the hatching rate. We need to look at that.
Thank you, Professor.
Thank you.
Hello, everyone. Yeah, maybe I have one. Hello. So thank you for the nice presentation, Professor. I would like to ask that, do you have any data on the chemical composition of these lakes?
Yes, data is there on these lakes. You have, I mean, Lake Texcoco, I haven't presented that aspect over here. Lake Texcoco is basically a soda lake with high concentrations of bicarbonates and carbonates and things. And there are other saline lakes like Lake Alchichica, which have mostly sulfates, calcium and magnesium sulfates. These lakes do not have very high concentrations of chlorides. So we used in our experiments a 1:1 ratio of bicarbonate and chloride to maintain the salinity during the study.
Yeah, thank you very much, because it was confusing for me that based on the literature, so a lot of publication refer to Lake Alchichica as soda lake, but there wasn't any data that's supporting this, that it has a high carbonate, bicarbonate content, or...
No, Alchichica has mostly sulfates.
Okay.
But this is not Lake Alchichica. This is Lake Texcoco.
Yeah. No. So you mentioned that this big lake Texcoco was once a big soda lake in Central America.
No, it wasn't a big soda lake. The whole lake was constructed. I mean, it was a huge lake which had sections which were freshwater and sections which are soda lakes. So the freshwater sections are, for instance, even now found in a huge system of canals, which covers almost, I think, 200 square kilometers over there at the south of the Mexico City. And it's a system of canals. That's freshwater. And in Lake Zumpango also is freshwater, but Lake Texcoco is saline.
I see. Thank you very much.
Thank you.
Do we have some more questions? No. Thank you, Dr. Salmon.
Thank you very much.
And the next speaker.
Yeah, if I'm correct, maybe I would be the next one.
Yes, that's correct, Attila.
Oh, okay. Thank you very much. Can I share my screen also?
Sure, go ahead.
Okay, just a moment. Yeah. So first of all, I would like to apologize for the quality of my presentation, because I have chosen a wrong pointer and I haven't got time to correct. So I would like to present a few figures that maybe it would be have better quality to visualize. So we have started to study soda lakes in Central Europe during my PhD. And So lakes looking like this, they are really shallow, flat, turbid, have a high pH, a lot of carbonate and bicarbonate dominating the water. And so they are extreme habitats for the aquatic life. And we were interested mostly about the bacteria community or the whole microbial community of these lakes. And we have a few seasonal data sets and also some functional investigation. But the main thing that during this time we have observed that these lakes has a really special microbial community comparing to other saline or brackish waters worldwide. And based on that, we would like to compare the communities of the soda lakes with other soda lakes from worldwide, which data are available, and also for saline lakes, for example, from Kazakhstan in this picture, you can see which has more or less the same pH and the same salt contact as salinity, but the ion content was different. And so we have started to collect data on that. Here you can see the distribution of soda lakes around the world. So we have talked about this central Mexican plateau, and also our main sampling site was the Carpathian Basin. And to collecting genomic data to identify bacteria based on a phylogenetical marker gene, the prokaryotic 16S RNA gene, I've tried to collect data from all of the Eurasian steppe, data from the East Africa Rift Valley, and data from soda lakes from America. And this is extended with other data sets from brackish environment, for example, the Baltic Sea, the Yucatan Peninsula. Also for Kazakhstan lakes, we had an expedition in there. And we would have liked to collect more data, but in the meantime, the pandemic came and all of our expedition was canceled. So if we compared overall more than 300 samples originating from 165 lakes, and there were some freshwater environments. So this is the bacterioplankton, the composition of the bacterioplankton of these shallow lakes. So you can see here freshwater lakes, here soda lakes from Eurasia, the Carpathian Basin, soda lakes from North America, Canada, and also from Africa. Among samples originating from marine waters, tropical seawater, from the North Sea or the Atlantic, extended by data sets with saline lakes found in the Kazakh steppe, also in Romania, from the Baltic Sea, some brackish environments. And as you can see that there is a clear distribution about that these soda lakes, the bacterial community of these soda lakes are mostly more similar to each other compared to other saline lakes, which dominated by chloride. And if I just highlight the salinity values in here, that you can see if when the salinity increases, then if low salinity, the type of ion doesn't seem to matter. So if it's a sodish environment, so these samples are from a big soda lake from Central Europe. These samples are brackish sites from river estuaries and also for some Baltic Sea and so on. So they are closely related, the microbial communities, to freshwater ones. But if the salinity increases, for example, here with 20 grams per liter, you can see a clear distinction between the communities of soda lakes and also the communities of saline lakes. And with increasing salinity, then you get communities of the saline lakes. So here's a few examples of the microbial groups characteristic for soda lakes in our investigations, and also other one characteristic for chloride-dominated saline lakes. There are cyanobacterial mats in some of the lakes, for example, for the soda lakes from Canada. That's why they are separated a little bit, characterized with this Nodoxylina species, and also traditional freshwater microbes characterizing freshwater environments, and brackish sites which has a salinity less than 1 gram per liter in most of the time. Thank you very much.
Thank you. Are there some questions? Are there some questions? Please ask. Nobody want to ask? Thank you, Attila.
Okay, thank you.
You made a good speech. Thank you. Please.
It is my turn.
Yes, that's correct.
Can I speak?
Yes, please.
Helle Mehmet, here. Are you with me, colleagues?
Yes, yes, yes. I can hear you. Would you like to present something briefly?
Yes.
Okay.
Dear colleagues, I believe or I hope that you have seen my slides, my presentation before, and I only underline some aspects. Our saline lakes are very, very fresh in comparison with your saline lakes. Baltic Sea is very brackish, and salinity in youngest coastal lagoons and open seawater are very, very similar. They are not different, but older lagoons are very fresh. There the salinity is only probably 1 milligram per liter or tenths of milligram per liter. They are near fresh. And what is interesting that they have formed during the whole Holocene in connection with different stages of Baltic Sea. And in existing coastal lagoons, nowadays existing coastal lagoons, there are some may be thousands of years old, and the youngest are probably 30 years old. And they are formed by neotectonic land uplift and with accumulation of sand. And some youngest lagoons are not real lagoons, they are very close. The vegetation is very close to freshwater eutrophic lakes. But old lagoons are especially interesting because they have developed freely during the thousands of years, and their human intervention has been not very remarkable. And in general, our lagoons are left by humans in general. It is good, and eutrophication has taken place mainly regarding the eutrophication of the Baltic Sea, because during the last 100 years or last 50 years, there was real eutrophication of the coastal areas of the sea because the fertilization from the agriculture was strong at this time. And occupation of reeds in lagoons developing during the last 50 years, there was very, very intensive and quick. And there is a large variety of species richness of lagoons. Old lagoons located probably 3 meters over the sea level and formed probably 3,000 years ago. They have very rich flora, very interesting and very many rare species. But younger lagoons, very very frequently they are covered with reeds, and the species diversity is quite low. Of course, these young lagoons with mighty reeds are also valuable for many birds and in other aspects. And we have not good ideas what to do for the future. We have thought that the story of, they are very temporary, the lagoons, but now it is clear that sea level has risen too. Separation of lagoons by uplift, it is some millimeters per year, but now sea level also rises some millimeters in year, and probably they are not so temporary, and we are able impact the future of our lagoons. Also, not it is only natural process of disappearing, but probably they are more permanent water bodies. But I can't speak so much. Please, questions.
Please ask who have questions. Thank you, Helen.
I am waiting for questions.
No questions?
No questions.
Perhaps we wait. Yeah, we just wait a little bit more. Maybe people. Yeah. So go ahead.
Question then. It's very nice, and a lot of lagoons you have been studying there, and you are mainly focused on the macrophytes. I know that's your major interest. But have you studied other things in the same lagoons, like phytoplankton, zooplankton, fish, and so on? Because it's part of the European, you can say, Water Framework Directive studies. So maybe there are other things also, like at least for fish and phytoplankton. Have you done that also? Do you have data from all these systems?
Yeah, yes, we have, and one book has published maybe 8 years ago in cooperation with other countries in our region. Yes, there are fish and birds, and relatively well studied are how to say, some 20 or 30 are more studied, but our study was aimed to find the criteria for the Natura areas. They are Natura area, Natura habitat, and how to estimate the value of lagoons, and there the macrophytes are the first factor what is important in Natura habitats. But yes, my colleagues have studied very many lagoons, but among 600 lagoons, of course, is not enough.
But have you quantitative data also for macrophytes, for instance, coverage and PVI, or have you studied that, or is it mainly presence absence?
Yes, of course. We tried in field works too to estimate how many percentage has covered with reeds and so on. Yes, and nowadays there are drones and so on, different possibilities to look them lagoons, of course. Yes.
Thank you.
Are there more questions? No, thank you.
Yeah, thank you all.
Thank you, Helen. Next speaker, please. It's
Ana Clara. Yes.
Hey guys. Can I start?
Yes, please.
Okay. Well, I won't show anything. I will just talk a little bit about my study. It is entitled Not Too Salty for Fish. The importance of variety salinity conditions for preserving fish diversity in eutrophical coastal lagoons. Well, in our study, we assessed spatial differences in abiotic conditions within and between three coastal lagoons in Rio de Janeiro. Actually, we had three average salinity profiles where we have a mesohaline system, a euhaline condition, and hyperhaline system. And then we assessed how these factors, these abiotic factors, influence the structure and composition of fish groups. We used guilds of trophic niche and also guilds of habitat use. What we found is that the lagoons differed in connections and the degrees of connections to the sea and also of freshwater input. So we had a gradient, how do I say that? We had difference in composition of both fish groups, and they reflect those, especially salinity conditions, because it was the main driver along with transparency and temperature. We also found that the speed of water renewal time was an important measure for the diversity. We had, for example, greater fish diversity and also the more different composition of fish groups at the euhaline system, because it was the fastest water renewal time, so it was very similar to the ocean. So we had a lot of typically marine species. And we also had the hyperhaline system, which was mainly dominated by brackish species. And the mesohaline conditions were dominated by, well, both groups, and it was the greatest, how do I say, typical groups. We had a lot of indicator species, so it was the most different, most dissimilar system. So we have the abiotic factors, something about the structural characteristics of the lagoons, about the connections to the sea and also freshwater input. And we can also, yes, question if there are also anthropogenic factors that influence those patterns. I don't know, I guess it was a bit confused, but I wanted to tell a bit of everything. So if you have any questions.
Thank you very much, Anna. Because I have a general question. So you have shown this variation in fish communities, right? Composition, so through space. I was wondering, how do you think we can expect climatic change in the future in these coastal areas to impact those fish communities in those systems? Because they are too close to the coast, right?
Yeah. We have some problems, some issues with those systems because they are near to populated cities. We can expect that they are really impacted by human activities, and we can expect, for example, that they get the connections to the sea get tilted, and you need to reopen them to renew the water. So we can expect that this needs to be done more often, for example, and that the absence of this or the decrease in the connections to the sea may increase also evaporation rates and change local abiotic conditions. And this can impact the composition and structure of the fish species.
Thank you very much, Anna.
Thank you.
Any more questions?
I also have a question. Hi, Anna. Congratulations for your work and presentation. My question is about the invasive species that you found. You mentioned the dominance of these invasive species in a system with more turbidity and higher temperatures and less salinity. So considering that species, invasive species, generally have a wide environmental tolerances, why do you think you didn't find in the other connected systems?
Yeah, we have found Nile tilapia, Oreochromis niloticus, at the mesohaline system. Well, it is actually a tolerant species, and we can find them in brackish waters. We have found in bays and other coastal systems here. But the other two systems, the other conditions, salinity conditions, are too harsh for them because they are cichlids. They have close past of marine species, but so they can endure marine conditions, but they can't endure too salty conditions, if you can. So if we have euhaline and hyperhaline conditions, it's too harsh for them. So I guess this is the only reason, because we have aquaculture facilities, those basins, freshwater basins, so connected to those lagoons. So we could, in theory, find Nile tilapia around these systems, but I guess that the salinity is something that limits their dispersion.
Great, thank you, Anna.
Are there more questions? No? Thank you, Anna Clara.
Thank you.
Next speaker, please.
Next, according to the list, it's you, Nickolai, the next speaker.
Yeah, but I have my presentation video. No video.
Yeah, so everybody had the time to watch it beforehand. So now it's more for general summary and discussions.
And no more participants.
So anyone has watched Nickolai presentation and has any questions to him? I was wondering.
Please.
So yeah, maybe because Eric already had some questions on stability right in the beginning. Maybe we could go to the next speaker. I don't know. If there are any questions, we can wait a few more seconds.
And why no my video?
Because your video was already in the Moodle system, so everybody could watch it beforehand.
Okay.
So that was the plan. I think everybody could already watch your video.
Yeah, okay. Yes. Next speaker, Luciana Neves dos Santos.
Is Luciano here? Perhaps he's not around. I don't see his name.
Okay. In this case, we have some time to general discussion, I think. Do we want somebody tell us something?
So maybe you can say a few concluding remarks for your session and new directions for investigation in the future, something like this.
All presentation were very interesting, and show us different aspects of saline lane ecology. And I think that now we have some lack in understanding of variable saline lakes, and only integrating all views we can go ahead. But I think that it's very difficult to talk about some general trends in development of saline lake study, except in one: it's connection of biogeochemistry of such lakes and organisms. But available data show us that it can be very interesting direction. And do you want somebody to add more?
So otherwise, I thank you very much, Professor Nickolai Shadrin, for organizing this session, and for everybody who contributed very interesting presentations. I have pasted the link for the ECR workshop that will take place this afternoon. So if you want to join, please use this general link that I pasted on the chat, right? So everybody is invited to go to the last day of early career workshop. And with this, I think we can conclude this session and have everybody a nice final days of the Shallow Lakes Conference, okay?
Thank you all. And in this case, we can finish our session. And I hope that everybody can write another using emails, and maybe it can generate new cooperations. Thank you all.